thanks for the invitation. Also, a big honor. So I think you do this all the time. You show the wall of fame, and then the people get nervous. They have to talk about. So I want to talk about threads, which is transfer into luminescent nanoprobe, and give you some examples. What we do with nanoparticles to do some biosensing. When I moved to Canada, and I think the first hockey game I was were the Canadians that had some animals, and I wanted to buy a t-shirt. $40. $40? And then I looked at the symbol and said, it's actually my initials, NHL, Niko Hildebrandt. So I adapted a little bit my logo, and now it's the Niko Hildebrandt. But I haven't been contacted by lawyers. So it's slightly different. But I thought NHL would work, and as every Canadian wants to be in the NHL, students are coming. So just to tell you where I'm from, I mentioned that already. So I was born and raised in northern Germany, so that is Hamburg here. It's almost Denmark. And for studying, I went to Berlin, and my PhD I did in a small city called Potsdam, which is very close to Berlin. And then I worked there as well in a Fraunhofer Institute afterwards, and then moved across Germany to France, where I worked at the University at that time it was called Institution up there. They like to change the name of universities and the logos all the time in Europe. But they're now the University. And then in 2019, so we stayed here for years, we moved closer to the sea in Normandy to Rouen, where I stayed for 4 years. Officially, I was professor there until 2023, but in 2021, I moved with my family to Korea, so National University, in the Department of Chemistry, which is a very different town than Rouen. So Rouen is, you see all of these old houses that are, none of the houses are straight. It's actually a very beautiful city. And Seoul is also a very beautiful city. I love Seoul because it's in the mountains, but much more modern. So if you ever think about doing some time in Korea, they have something that's called the Brain Pool Program. When I first saw this, I thought I'd bring a brain. It's the program, but it's not like this. They just invite people from outside, and it's funded by the National Research Foundation of if you want any information on that, I'm to share that. And well, you know that little city here, Hamilton, that's where I'm now. They say, well, I don't think I have to I have lived in Hamilton for a month, and then I moved. So there, when I think about ways of understanding life with nanotechnology, there were some very early ideas. So some of you still might know the movie of Richard Fleischer, 1966. I wasn't even born then, but a fantastic voyage where they shrink scientists down and they go into the body and find out all kinds of different things. So obviously that doesn't work very well, but the idea is great. Some of you might know this, the Waterloo. I don't know if they show it in Canada, France, and Europe. So where they have also these, it's actually how the body functions, and all of them are small figures in the body, and they also have the same principle as Richard Fleischer did. It also doesn't work, but what works, in our case, at least to some extent, whatever physics is to combine luminescent nanoparticles and biohybrids with a research team. So I like to use this picture of the research team as Captain Future. I even stole Captain Future's sign because he's got an F sign for future to make it a threat sign. But this is a manga from the 80s, but it was, so Captain Future was invented by Edmund Hamilton. So that's why I came back to Hamilton, because of research in Hamilton. And actually, he's also a great superhero because Captain Future is a scientist. He doesn't have superpowers, he just has a laser gun, and he's got a very cool team. He's actually what I would say the perfect image of a professor. He takes all the things, but the group does all the work. So he's always into some trouble, and then his group saves him. He's got a flying brain professor. This is Professor Simon. Well, I could talk about Captain Future for 45 minutes if you want, but I recommend to watch it. So, if we have these nanomaterials or molecular nanomaterials, and we have the interactions with light, it can really help us in understanding and making things better on a sensitivity and resolution basis, and that is where Fred comes in. Fred, or first resonance energy transfer, about these molecular or nano light interactions and how these interactions change over the distance. So in a nutshell, RET, or first resonance energy transfer, fluorescence resonance energy transfer, is an energy transfer from a donor to an acceptor, and it can be modeled as dipole-dipole. Transition dipole moment of the donor, which is the emission of the donor, and you need a transition dipole moment of an acceptor, which would be the absorption of the acceptor, but it's not emission reabsorption, it's really the interaction of two dipoles, which is 1 over r to the 3, but as they both interact together, it's 1 over r to the 6, and they need to be close together in a good orientation. You can imagine like the magnetic field or electromagnetic field around the donor. They are like this, right? And the acceptor has to be oriented in a nice way so that the energy can be transferred. So you need close distance, 1 to 20 nanometers, and you need resonance, energetic resonance. So the emission of the donor needs to have the same energy as the absorption of the acceptor. And all this discovery goes way back to the 20s, Frank and Carido, and in France, and in London, Germany, a lot of discoveries about these interactions. And we like to call it first resonance energy transfer because Theodore First was really the person who had the best relationship between easily accessible spectroscopic data, so emission and absorption, and the theoretical equation so that this could actually be used in some applications. So, as I said, it's a resonant overlap, so the resonant energy transfer means the emission of the donor needs to overlap with the absorption of the acceptor, and the efficiency is 1 over r to the 6, r is the distance. So you have something which is called the first radius, this is r naught, r0. This is where Fred is 50% efficient, and that's around 3-4 nanometers. You can see, taking the example of 4 nanometer, 50%, and half of that, so at 2%, so at 2 nanometers, you're at 100%, and at 1.5 this, so at 6 nanometers, you're almost at 0%. That means between 2 and 6 nanometers, you're going from 100% to 0%, which means that in this steep region here, you have a resolution which can be easily set nanometers. Just by doing spectroscopy, and we can do quantum yields, but I would recommend that because they're very hard to measure precisely and accurately. But if you take luminescence intensities, or luminescence decay times or lifetimes, and you do 2 measurements, the donor and the presence of the acceptor and the donor alone, and you find the Fred efficiency, and then with this formula you can find the distance between them, and you only need spectroscopy. So many people call this the first optical super resolution technology before all these other super resolution technologies with microscopes came up, and it's even much more super resolution than super resolution microscopes, for example. Optic ones, maybe 10, maybe 20 nanometer resolution. With Fred, you are always in the 1 to 20. You can go larger because afterwards it's not efficient anymore, but in this range you can measure interactions pretty well. I also like it because Fred has been cool for more than 80 years. I don't know if you know David Bailey, but he's a photographer in the 90s in London. I was in working in art gallery, and I bought this poster of Michael Caine, who is an actor, so young people should know him. That's the servant of Batman, Superman? Batman. But, well, he also already existed in the 60s. And I pasted in this picture of Theodore First from the 40s. And I'm not encouraging smoking to anyone, right? But at those times, smoking was considered really cool. And I really like this poster of First the lab, he's got a lab coat and doing some smoking right there. Luckily, that's not allowed anymore these days. So the different types of resonant energy transfer, we can name them all Fred if we want. but it really depends on if the dipoles are point dipoles or if they're surface dipoles or volume dipoles. So in the classic approach where you have two dyes, they can be easily seen as point dipoles, and you have a 1 of r to the 6th relation. We call this usually Fred. Then you can also have energy transfer to gold nanoparticles, because gold nanoparticles have a strong plasma band in the, well, usually green, so around 530 nanometers, and they can quench fluorescence very well. A lot of people call it Fred, but it's actually not Fred, it's also rather than energy transfer, but as point dipole interacts with surface dipole, the nanoparticle is nano, but it's so big compared to a molecule, the dipole of the donor interacts with many dipoles on the surface. If you calculate that with a simple formula, you come to an energy transfer efficiency of 1 over r to the 4, which is actually not bad because you can have larger distances. For Fred, we have maybe 10 nanometers. With NSET, what we usually call a nanosurface energy transfer, you have distances of up to 20 or 30 nanometers. So here are some classic examples of Fred. So fluorescein as a donor, rhodamine as an acceptor, so two fluorescent dyes. You see the emission, you excite fluorescein here for 50 nanometers. The emission of fluorescein overlaps with the absorption of rhodamine, which means rhodamine can get excited, you have emission of rhodamine. And by now, also a classic example are fluorescent proteins. And fluorescent proteins are often designed for sensors, sensing calcium or other ions. So you have a sensor ligand domain here that is open and closed. And on the two ends of the protein, there's a CFP, which is a signed fluorescent protein, and YFP. The far apart, the Fred efficiency is low, and if they're close together, the Fred efficiency is high. And the classic NSET example is gold nanoparticles. So you have the energy transfer from the dye to the surface of the energy transfer of the gold nanoparticle. And that is also important because the nanoparticle is relatively large. So if you have a 20 nanometer gold nanoparticle, you cannot calculate this simply from the center because the dipoles are not here. The surface plasmons are on the surface, so the dipoles are here. So the energy transfer is always from the dye or from the terbium complex or whatever to the surface of the gold nanoparticle. It doesn't matter how big the nanoparticle is, it will always be the same from this center of the dye or the complex to the surface of the fiber or to the surface of the 18 nanometer. In our lab, we like to work with lanthanides, with lanthanide complexes, so this could be europium or terbium complexes because they emit the visible range. Otherwise, lanthanides are all over. Everybody's got a lot of lanthanides in their pockets. At least if you're wearing a smartphone in your pocket, the light here is lanthanides. The magnets in your cars are of lanthanides. And I think 15 years ago, there was a big crisis of lanthanides. Because the problem with lanthanides is they're very difficult to extract because they're all 4 elements and they're very similar. So distinguishing between europium, dysprosium, neodymium. Neodymium is used a lot in all these magnets. And so a lot of mines in North America and Europe closed. And in one moment, China had all of the mines. And then they said, because Chinese people are smart people, they said, well, if we have the monopoly for it, why shouldn't we increase the prices by 2,000%? That's what they did, is get a big international crisis, not because I can't do any more fluorescence imaging, because we don't need a lot for that, but because the whole car industry would have to shut down, because they all changed from the big magnets that you have in your speakers, because we don't only want to drive, we want to listen to music. And so the speakers that use iron magnets are that big, and with lanthanides are that big. So they would have to change the whole industry of building doors, and then I think they could lower the prices again. And now you can see that, especially also because of smartphones and magnets and windmills, that several lines for lanthanides come back up in North America as well as some in Europe. For us, we need very low amounts, so some milligrams or micrograms, so we don't care much where it comes from. And if we have a milligram or two, we can work with luminescence almost the whole of our lives. And we put it also in nanoparticles, up-conversion nanoparticles, for example, and then make energy transfer to other nanoparticles, which could be quantum dots dyes of frozen proteins, and sometimes even from the dye from lanthanide to a column dot to a dye to another dye. We're having multiple energy transfer steps, sometimes even with useful applications, sometimes we're showing that it works for the fun of it. But sometimes these multiple energy transfer steps, so these kind of photonic wires, have actual applications. In sensing and with Fred, what we want to do is measure biological interaction. One very well-known or long-known application are these homogeneous liquid phase immunoassays. Homogeneous means these are mix and measure assays. You just put it into a solution and you can measure it without any separation, which is the opposite of an ELISA, which is a standard assay in biology where you immobilize an antibody on a solid surface, and you wash, then you bring the biomarker that you want to measure in, you wash again, and you have a second antibody, then you wash again, then you put your application peroxidase inside the powder, and that takes a lot of steps. So here we mix and measure because of the energy transfer. So you can see these assays, they have commercial names like CR-FRED, HRF, Trace, NANS. They are a lot of plate readers, so if you're using plate readers, you often have an option for HRF or HRF. So what you have in the case of an immunoassay is two antibodies, and these two antibodies are specific for any kind of biomarker. So in this picture here, you don't have the biomarker, but you have the assay kit, and what you have is a UV excitation, which will excite the lanthanide, labeled to this antibody, and you will have this long millisecond lifetime emission, because lanthanides don't have normal fluorescence, so it's not nanoseconds like a quantum dot or dye, but as they have, as they're 4F elements, they have very forbidden transition states. So the absorption is very bad, and the emission takes a long time, and it's usually also very bad. So they convince a lanthanide for a long time to emit. That means their emission is in the millisecond range. If you also excite directly the quantum dot or the dye on this other nanobody, you can see that there's very strong but very short emission because this is usually nanosecond. What you also have if you work with cells or with animals or with serum or blood is a lot of background fluorescence. We call that autofluorescence, and that is in the nanosecond to microsecond range, symbolized as brown bar. So now you add the biomarker, or usually what you add your assay kit to blood sample or serum sample, and then both antibodies will bind to it. That's what we call also a sandwich immunoassay because they form a sandwich. So my head is the biomarker, and these are antibodies. Imagine a sandwich between the two of them, and that brings them in close proximity. So now this lanthanide is close to the dye, say 10 nanometers, and what can happen is Fred, energy transfer. And if you remember the Fred efficiency equation I had a few slides ago, you have to measure the lifetime of the donor in the presence of the acceptor and only of the donor. That means if there's energy transfer, the lifetime goes down. So you have the emission of lanthanide here. When it's quenched, the intensity goes down and the lifetime goes down. But what's more important is that now the energy that the dye or the quantum dot gets is not the direct excitation by light, well, that is still there, but it also gets excited by the long states of the lanthanide. So the dye or quantum dot gets excited, gets de-excited after a nanosecond, but the lanthanide is still in the excited state for milliseconds. So after a few nanoseconds, you can easily say that 99.999% of the beryllium or the europium is still in the excited state, and then you can have energy transfer, and that means that the dye or quantum dot gets the same long lifetime than the quenched lanthanide. So all this orange here, the emission, the long lifetime, is only there if we have this biomarker, because before it wasn't there. So that is our separation. We don't need any separation by washing. We just do separation by Fred. What we need then is time gating, so we use a flash lamp or laser. So this is milliseconds. That means we go up to maybe 100 hertz or kilohertz. So that can be easily done by a flash lamp. And then we only measure the intensity that appears after a few microseconds. So in this case, for example, between 500 microseconds and 2,000 microseconds, and you can see there is no background from the autofluorescence. So without any separation, just by separating at the lifetimes, we can have zero background. And well, we know, unfortunately, zero background doesn't exist. There's always a little background, but very low background measurement without any separation. And you can do this on many readers. So these are just some examples of readers that a lot of people have in their labs from different companies. We like to use this system, which is a clinical plate reader, Cryptor, by Thermo Fisher. We develop also our own readers that can do lifetimes. So we call it the green reader. You can also develop your own time-gated microscopes with optical choppers. So all this is on the millisecond and up to kilohertz range. You can easily do this by choppers you buy off the shelf from all labs. Or you can also use ICC cameras. They're a little more expensive, but it's the same system that you're gaining the intensity over time. Two things we like to do with red, or two other things apart from getting rid of our autofluorescence, is multiplexing to measure several biological interactions simultaneously. That can be done by counter multiplexing, so we excite one lanthanide, so for example terbium, and as the emission is quite broad with several emission lines, you can make energy transfer to different acceptors. So we can use the same terbium donor and use 3 different bi or quantum acceptors to measure 3 different DNAs. And for simplicity reasons, I show the same DNA here, but just imagine these DNAs different. You need only one excitation of the donor, and then you measure 3 different colors of receptors and measure 3 different DNAs. But we can also do time multiplexing because the quenching of the lifetime of the donor or the sensitized acceptor depends on the distance. So now imagine you have the donor here on the DNA and the acceptor here. Then you move the acceptor on the DNA, move it by 0.3 nanometers because each base pair is separated by 0.3, 0.4 nanometers. You can adapt this distance, and then the quenching will be different because the donor and the acceptor are at different distances. And here's an example of different intensities. I think it's 3 different distances, and then you get a multi-exponential decay. So 1, 2, and 3 exponentials. Now you have 3 different time windows. So you excite once with your laser, and you measure here, here, and here. You correlate these windows, and then you can measure 3 different biomarkers of 3 different DNAs with one single frame there, just by exploiting the time component of fluorescence. So what kind of applications can you do with this? And I want to show you really maybe 4 examples about using different nano approaches of measuring different molecules. First will be about FRET immunoassay. So that's the same principle as I showed before. We use antibodies for it, and we can combine terbium antibodies with dye antibodies, or we can combine terbium antibodies and quantum dot antibodies. If you're looking at quantum dots, so semiconductor nanoparticles, they're great because you can tune their emission and also their absorption simply by their size and also by the material. So we like to work with cadmium, sulfonide, cores, tin sulfide shell quantum dots, not because cadmium is so nicely toxic and we like to spread it all over a biological system, but just because they're the best quantum dots you can get from the point of view of the photophysical properties. Usually they're well protected as well, so they don't do a lot of harm to the body if they go inside and outside. I always tell the students, you always have to be careful if you read studies where they put nanoparticles inside an animal, and then they measure the urine or something, and they say, well, here they are outside, so it means it went in the body. Without the body, there's no harm. Then I usually give the example, it's the same as I would take a knife, stick it into your body, it's in the body, it's out of the body, here it is, it did no harm. It doesn't mean if it goes in and out that it does no harm to the body, right? So it can still be harm inside, so you have to be careful about that. We like to use it for immunoassays. They're usually outside of the body because we take a serum sample, so there's not a big issue, and the amount of cadmium you're using is also. That just as a side remark, what I wanted to show here is that size matters, because FRET is distance dependent, and FRET works in a range of about 1 to 15 nanometers, and the quantum dot already has, if you see it as a spherical quantum dot, then they're often not spherical, and they have a size of 10 meters on their own, 10 nanometers on their own array. Then a full IgG antibody from top to bottom is also about 10 nanometers. Now imagine you use the same scheme that I showed before, 10 nanometers plus 10 nanometers plus an biomarker plus another 10 nanometers of antibody plus the acceptor. That would mean the donor and the acceptor are about 30 nanometers apart. That won't work for FRET. And what we do usually is we reduce our antibodies. So we cut it here, we cut it there, so we make them smaller, that we get them closer to the quantum dot, or we can also sometimes orient them differently on the surface. Or we use artificial binding proteins, antibodies, or like this albumin-derived proteins, or these repetitive proteins that can also recognize, similar to aptamers. So you look for many different of these proteins and see how they bind. Then you can get them very close to quantum. So one study a few years ago, we showed that these nanobodies can be attached with different standard chemical techniques, so like with histidine tags, or the biotin tag, or the cis tag, to different commercial quantum dots. And these quantum dots had small ligands, so this is a CL4 ligand, so it's just a very thin coating of a ligand on the zinc sulfide shell, and then this his tag can self-assemble to the zinc on the quantum dot. A few bi-quantum dots with streptavidine on their surface, so we can buy those off the shelf at several different companies. And you have a biotin-related nanobody, you can get them with quantum dots. Or if you have large packed chains for protecting your quantum dot around it, you can also functionalize cysteine-tagged nanobodies and put them on there. And on another nanobody, so this would be the acceptor nanobodies on the nanoparticle. On the other side, we put a terbium, so lanthanide on this nanobody. Both of these nanobodies are specific for EGFR, epidermal growth factor receptor. And when we add the EGFR to it, we have this sandwich complex. Now the sandwich complex looks a bit different because it's around the nanoparticle. Right? So per quantum dot, we have here I could show 4, but probably we have up to 20 different energy transfer possible. We can see that these are the usual calibration curves that we established. So here's the concentration of our biomarker, which is serum-derived epidermal growth factor receptor. This is the relative FRET ratio, so that's the time-gated intensity of the acceptor divided by the time-gated intensity of the donor. So it's your FRET intensity, if you like. You like to use ratios because they give you a higher accuracy of your measurements, and you can see that this FRET goes up with the concentration of the biomarker. So you can measure those at low nanomolar or picomolar concentrations, which is nice because you can do it at different colors, and you could possibly do multiplexing. But so that was nice work showing that it works with the quantum dots. But the surprising thing was that luckily one of my students at that time mixed a terbium nanobody that also had a histidine tag. We usually don't want a histidine tag on it because this is not supposed to bind here. Right? It's supposed to bind to EGFR, and this one is supposed to bind to the quantum dot. And so, well, this is the proof that sometimes students should just play around with things, and sometimes also the proof that if you're mixing different things by error, you can find very surprisingly nice things in research. So we didn't know for 2 years what it was because we always saw that the signal didn't work because we wanted to do these sandwich assays. But when I finally found out that, oh, possibly this terbium is binding to this quantum dot, it's maybe displaced by the EGFR. We left out the other nanobody and labeled the terbium with the histidine here, and then added the EGFR and what the EGFR does, and surprisingly only to this nanobody and not to the other nanobody. This is just because the binding sites to EGFR are in different places of the nanobody, or no, the nanobodies bind to different places on the EGFR. So for this one, EGFR displaces it away. So that means the FRET intensity goes down with the concentration of our biomarker, and we can establish very sensitive, so we can go down to 80 picomolar in the limit of detection, but highly simplified because you only need one antibody or one nanobody, you still have a radiometric measurement, so it means less reagents, it's simpler for storage, and it's also much simpler for presentation. Second example I want to talk about is the detection of DNA and RNA. So also for clinical applications, measuring microRNAs and measuring cell-free DNA in the circulating blood. So for doing this, we had to go away from the simplification because, as I mentioned before, we like to do simple assays where we just mix and measure. And we can measure things at picomolar concentration, which is great from an analytical point of view. But if you want to measure circulating DNAs or microRNAs, they are very low in concentration, so picomolar or maybe even lower. And you don't want to develop an assay. I mean, we develop these assays called biosensors, but if you talk to a clinician, they say, well, if I measure the microRNA 21 or let's say 8 or different things at nanomolar concentrations, that maybe means that the patient is already dead. I don't want to measure a lot of a dead patient. I want to measure things early in advance. So we can have to come up with something that amplifies our signal. And the great thing about nucleic acids is that they can be amplified. Everybody knows PCR. But PCR is problematic because you have heating cycles, right? You heat up to take the DNA apart, then you amplify it, you cool it down, so you have the thermal cycle. And that's not easily adaptable. So we were looking for some isothermal amplification. We came up with this principle that people had developed a long time ago, which is rolling circle amplification. So we have a DNA, which we call a padlock DNA. So it's called a padlock because it's a single-stranded DNA, and the two ends bind the target. The target is here, it could be DNA or RNA, and once it hybridizes, it closes like a padlock. That's why it's called a padlock probe. And once you have the target hybridized here, you need to close this open DNA here to a ring. So you can do this with a ligase. So you ligate this open nick over the DNA or RNA target split. And then you have a single-stranded DNA on a closed DNA ring. And what a polymerase does, if it sees it, the polymerase looks here and says, this DNA is not complete. I want to complete it, so you put the NTPs and the polymerase inside. What the polymerase will do, it starts here and synthesizes new DNA around the string to make a double string. And I have always called it a stupid polymerase because once it goes here, it doesn't stop and say, finished, it's done, I did my work, now I go for coffee, then it continues. So it displaces the strand that it created first, and does it again, and does it again, and does it again, and does it again, and does it about 1,000 times until it stops on its own or you stop it. What that means is you create a long rolling circle product which always has the same DNA sequence, right? Because you're always replicating this sequence once, twice, 3 times, 1,000 times. So that means for one target, you have a single strand with 1,000 times the same sequence. And now we take our friend probes, so there's the donor here in green and the acceptor in red, and as the sequence repeats again and again, we can design them that they're specifically oriented on this strand that the rolling circle product created. And if we have this, we can exploit two things. First, we can use different colors. So this is a spectrum of a terbium donor. So this is a lanthanite with these different emission lines. And it overlaps with the absorption of psi 3.5 and psi 5.5, so you can do energy transfer, and you can make 2-color multiplexing. But now we can also create our rolling circle padlocks with different sequences. So that means we can design our DNA probes, or single-stranded donor acceptor probes, to bind or to hybridize at different distances. You see here, for example, we have 10 base pair distance, and here we have 18 base pair distance, which means we get different decay times. So you can see this is a multi-exponential decay of a donor, and here we have this multi-exponential decay. So it means we can combine, in this case, 2 colors with 2 lifetimes and make 4-fold multiplexing. So we can measure 4 DNAs with 1 measurement from a single sample. That works very well from the sensitivity point of view, so we can measure all these different DNAs at limits of detection that go down to attomolar. So 300 attomolar would be 40 zeptomole. Below zepto, there's only yepto, and then there's nothing. It sounds very low. It's also low, but you still have to imagine by Avogadro's constant that 40 zeptomole is still 24,000 microRNA. So we're not measuring a single microRNA, although it is pretty low. Now we have to do these messy calibration curves, so we have to calibrate for all of 4 time gates, and then our assays, different samples get even messier, so we have 10 different samples with all the 4 DNAs inside at different concentrations, which we know, so these are these dotted lines, and we can find those quite well to recover these at low picomolar or subpicomolar concentration. And do the same, so this one was DNA, we always start with DNA for developing the biosensors because DNA is cheaper, but you can also do it with RNA because RNA also hybridizes to DNA and measure, for example, microRNA 21 in plasma samples or in tissue samples, here of colon breast cancer, the plasma of ovarian cancer, or we can measure different microRNAs like microRNA 132 and 46 in different cell lines to analyze it. Or we can also measure circulating tumor DNA. It's a bit more challenging because unfortunately DNA is always double-stranded. And what you don't see in a lot of papers is when the people develop the sensors, they always sense single-stranded DNA. But single-stranded DNA is very rare in your body, so that means you have to take it apart. Denaturating DNA is not a problem, but if you want, so you denaturate it here, and then you want to hybridize it to your padlock. Your padlock has the same sequence as the other single-stranded, right? Because it should hybridize. So forgetting this step is actually, well, it's not cheating, but it's going far further away from reality because for sure you're losing at least a factor of 10 or 20 in sensitivity because you need to denaturate your DNA. But it still works pretty well. So we have picomolar concentrations that we can still measure. And we can measure the mutated DNA, so with the single point mutation here compared to the wild type DNA. And we can even turn the whole thing around. So a colleague came to me and said, well, can it also measure the DNTPs? So what the polymerase does to synthesize DNA, but these DNTPs could be important in cells for measuring treatment with different drugs. So we used our rolling circle amplification. So this, how we were interested in the target, we designed it ourselves. And then we stopped at the ATP, the TTP, the CTP, or the GTP at one moment. And then we could know the concentrations of the other because they always stop when the fuel is empty. We measure these things. One thing from the nano point of view is that you always need to be careful if you want to combine different nanosystems. And so for this paper, I designed this table of contents, and it wasn't even accepted by NanoLetter, so I think I have one of the nicest table of contents images that exist in NanoLetters, from my point of view, because I do it myself. But the idea was so that the student I had, she wanted to design this rolling circle amplification thing on a quantum dot, because quantum dots are great for multiplexing. But what it means is that you have a quantum dot, and they call it the QD cat, which is about, let's say, 20 nanometers, maybe 10, maybe 25. I have fret, which is also nanometric. That's why I call it fret fly, which is about 5 nanometers. And then I have the amplification product, the rolling circle amplification, and I call it the DNA amplifier. And what, so it's very easy, like a lot of problems are easy on a whiteboard or PowerPoint, right? You mix it in the beaker, and then you get the QD fret DNA fly cat done. But for sure, it's not that easy, and so I won't tell the whole story, but over 3 or 4 years, the student tried different things she's read in the literature, even with quantum dots, circling here, cutting at different things, putting different ligases and polymerizes inside, recycling the target, cutting on the product, cutting after the product, and even cutting twice on the product didn't work. So in the end, she came up with a double amplification, which was relatively complicated, and it worked very nicely, but it worked as well as it worked with the dots. So it means all these things done, which are very interesting from a point of view. So I think the supporting information of this paper is probably the most important. It because a lot of people can read it and say, don't try this, don't try this, don't try that, because a lot of researchers try the same thing and fail because nothing of it is published before, because we don't publish things that don't work. And here a lot of things don't work. A little bit more about imaging. So we also adapted quantum dots for imaging in cells or in live animals. One thing we were interested in was the development of zebrafish larvae. First of all, because zebrafish larvae are great data transferring, so you can image them very well, and because their development is very quick. So from one cell to like almost a fish already, it only takes 24 hours. So you can see this development and wanted to see if we inject quantum dots with terbium, so for this time dating, in a one cell stage, can we still see it after 3 days when there are thousands of millions? So the probe we used is a quantum dot that has again this terbium on it with a peptide, but now it's always attached to it for the histidine tag. So we have terbiums here. We also attach a maltose binding protein. This is just simpler for microinjecting because quantum dots like to clog your needles with your microinjector. So if you microinject it and you have some proteins on there, it goes easier. And we also have some space that fluorescent proteins that are expressed in the cell or in the animal. And if we express them with a histidine tag, they will self-assemble in the cell to this quantum dot, in which we have energy transfer from our terbium emission to these 3 different quantum dots. So we use 3 colors: turquoise, orange, and red. So the energy transfer steps here will be terbium to the 3 quantum dots, also terbium to the fluorescent protein if it binds to the quantum dot, but also this quantum dot can transfer energy to the fluorescent protein. So these are the multiple energy transfer steps that you can have. And the idea is, as before, we excite, the background is over after, say, 10 microseconds. And afterwards, we only measure here the background free intensity of our terbium, the quantum dots, and the cherry fluorescent protein. And here is much more important because the background is really strong in animals and live animals, much stronger than in the buffer, for example. We tried it first with cells, where we could see we could make energy transfer from one terbium donor to the 3 different quantum dot acceptors. So we got different colored probes. We could also see that in cells that expressed this cherry fluorescent protein, so these 3 here expressed it, this one doesn't. So sometimes it's nice that not all cells express the protein that you want. And if you get them on one image, you can show with one image that you have energy transfer, so from the terbium to the quantum dot to the fluorescent protein only where it's expressed. You can see here we have the quantum dot and the terbium inside that cell, but we have no cherry emission because there's no cherry expressed inside there. You can also do the control experiment where you see it only with the quantum dot and the cherry, and you see there's no energy transfer. Then if we go to the zebrafish larvae, you can see if we make time dating, and we have here the injection at the 1 cell stage and measure after 4 hours, 24 hours, and 72 hours. You see after 72 hours, you have to squeeze the zebrafish larvae in a channel. So they're not actually that elongated, we force them inside, they don't move around. I wouldn't say they're happy, but they're alive. And you can see we have the donor emission and the acceptor emission. We have no background because it's time dated. So if we do the same on zebrafish larvae without any injection, we see that after 72 hours, the red background appears a little, but we can see if we do this without time dating, so we excite with a continuous wave. But in the green channel, the background is exactly the same as the sensing that we want to do, and even in the red, it's much stronger. So this time dating, getting rid of the background really works, and it also works after several hours. And we only inject once. We inject in the 1 cell stage. But every separation, the quantum dots don't get more, right? They're always the same. They're in other cells, but they're still bright enough to do the sensing. So we can do this over 168 hours. That's about 5, 6, 7 days, maybe. You can still see it. You see the exciter gets a little nice, and you can see there's almost no background in the. Last thing is upconversion that I want to talk about. And upconversion is great because you can transfer NIR light into visible light. And this also works with lanthanides because lanthanides, again, have these long-lived excited states. So you don't do two photon excitation, so what you can also do with a femtosecond laser, then you have two photons that arrive at the same time at your molecule. That puts them into a not real state, but into an interim state that is very short-lived, so you can get two low-energy photons and get one high-energy photon out. But for lanthanides, you don't need this because these states have a lifetime of several microseconds. So you put in one NIR photon, then you put another second NIR photon inside and get blue, green, or red light out of these lanthanides. Very often it's a combination, so you have sensitizers like terbium, and you have activators like thulium and erbium. You can see here, so all of these samples are excited in NIR, so lanthanide, you can see, and what comes out are different colors in the visible. So erbium would be this is different concentrations. Terbium would be more in the blue and less in the red, and if you mix them together, you can get everything. So we wanted to use these upconversions for making theranostics where we excite in the NIR, and here it's interesting because if you want to do theranostics in the body, or at least below the skin, a lot of people say we use upconversion for deep tissue imaging, which is true because you can define deep like you want. If I say 2 nanometers is deep, well, then 2 nanometers is deep tissue imaging. But I think if you talk to people in general, deep tissue imaging would be something like X-ray. Really go deep into the tissue. Optical deep tissue imaging means a few millimeters. You can go below, but you cannot do it with visible light. See, the green laser pointer, you don't see it here. Believe me, it's on. But a red laser pointer, you would see it there because red light penetrates better, but it's also scattering, and NIR light even penetrates. So if we use our NIR upconverted nanoparticles, we could excite them at least below the skin. The idea here is that we have photosensitizers, so they produce singlet oxygen to destroy cancer or other cells, targeting for cancer, but these need to be excited in the visible. So they have UV or visible excitation, which means you cannot excite them where you want them to be excited. But if we encapsulate them on the surface of our upconversion nanoparticle, we excite our upconversion nanoparticle with 980 or 808 nanometers, which is how much can penetrate. Then these nanoparticles will create a UV or visible emission, this black spectrum that you can see here. And that overlaps with the absorption of these different photosensitizers that you can see here in the visible. So we excite here, the energy is transferred here, which is pentaplate re-excited, and then you can transfer the oxygen into singlet oxygen and destroy your cells. And at the same time, you can follow it because you have the emission. So that's what they call theranostics. So we have the therapy and the diagnostics at the same time because we can see what's happening in the cells with a microscope. So you want to be sure that they are not released without photoactivation. So you can see that over several tens of hours, we still have a release of about 3-4%, which is okay because more than 90% of the photosensitizers stay inside. And then we also want to see that singlet oxygen is created, and if we excite our HCNPs, we can see the singlet oxygen phosphorescence, so nice oxygen phosphorescence, which is not very strong, but you can see it in the NIR. So we can see that singlet oxygen is actually produced. And you can see here the cytotoxicity assays with, I guess, a HeLa cell line, I can't remember. We have no light. There's a little bit of toxicity with high concentrations of those nanoparticles, but not a lot. But if we excite, you can see that we kill almost 100%, at least 30%. We did it at different intensities because this intensity is really strong. So it's 3.2 watts per square centimeter. And a lot of people, when we say, we do the lanthanide complexes, we do imaging with it, they say, well, that's UV light, is that dangerous for cells? I said, yeah, but we excited kilohertz or something, so the dose is really low. Nobody ever complains if you take an NIR laser, because you can't see NIR, and you shine 3.2 watts per square centimeter. It gets really hot, so when there are regulations, and a lot of papers in nano journals where you can see in vivo experiments, because you want to publish your work in I don't know, advanced materials or something. The science is important, but what is more important, that you can do your stuff in a live animal. Because, I don't know, maybe they all grew up, the editors in slaughterhouses, and like to kill animals or whatever. Sometimes I would like to see more characterization of the materials before you put it in any animals. I'm not against animal testing. You need this before you put medication into humans. But this really burns the skin. But they never show the skin. They just show the effect inside. They said, well, we killed the cancer. I said, well, the easiest way of killing the cancer is just taking the mouse and put it in solvent or in acid, right? The cancer is there. The mouse as well. But so this is way too strong. So we have to go to lower intensities. And still see that there's an effect inside. And so the summary of this is we did different live depth fluorescence imaging on cells. We also did some sensitive probes, and we can even see that the cells are deforming at both intensities, so we can see that the cells are actually killed with illumination only and not without illumination. We also did some single nanoparticle cell barcoding. If you're interested into that, I'll be happy to discuss this. Some enzyme kinetics and molecular logic gates. So we're not building computers, but we're building molecular logic gates and or NAND inhibits with this energy transfer processes. And we also used FRET as a molecular ruler because in the beginning, I always start my introduction with you can measure distances and then I never show anything. But you can use FRET also for measuring distances and conformations. So with proteins or nanoparticles or DNA, we can see how the binding or opening and closing of proteins or hybridization of DNA on these nanosurfaces works and changes. I'd like to thank all our fantastic collaborators who work with this over the world, and for sure the funders for the money they gave us over the years. I'd like to thank the people who worked on it in the different groups over the years in France and Korea and Canada. This is my current group at McMaster University, and I'd like to thank you for your attention. If you're ever in France, because in France, FRET just means transport, and they have these trains, so look out for the train. When I came from Germany to France, and FRET over 10 nanometers, that these French people, they do FRET over thousands of kilometers, right? Because they go from Lille to Marseille, which is about 1,000 kilometers, and they still exist. So look out, they have a FRET train there.